The discovery that the genome encodes all these regulatory RNAs means that a vast portion of the transcribed genome serves regulatory rather than protein coding functions. Mutations affecting these non-coding RNAs can cause disease just as readily as protein coding mutations. A child with autism might carry a mutation disrupting an LNC RNA essential for neural development. A cancer patient's tumor might have amplified a circRNA that sequesters tumor suppressor targeting miRNAs. Structural variations and the missing heritability. Alongside understanding regulatory elements, researchers have recognized that the genome is far more structurally variable than For decades, geneticists focused almost entirely on single nucleotide polymorphisms, SNPs, which are common genetic variants, affecting individual DNA letters. Yet most humans carry hundreds of structural variations in their genomes, some inherited, some arising de novo. Many structural variations are benign, but others profoundly affect phenotypes. A duplication of a critical developmental gene might cause intellectual disability or autism. An inversion might disrupt a regulatory element and cause disease. Importantly, by 2028, researchers have realized that structural variations account for a substantial portion of the missing heritability that plagued genetic studies. For decades, genome-wide association studies, GWAS, identified genetic variants associated with diseases and traits. Yet these variants explained only a fraction of the estimated heritability, the portion of trait variation attributable to genetic differences. Researchers term this discrepancy missing heritability. One major contributor to this missing heritability turned out to be structural variations, which older sequencing technologies had largely missed. Advanced long-read sequencing technologies developed in the late 2010s and refined through the 2020s can now detect structural variations with high precision. Pan-genome projects, which sequence not just a reference genome but diverse individuals from many populations, have revealed that structural variation is far more common and consequential than previously appreciated. Some individuals carry dozens of structural variants relative to the reference genome, affecting hundreds of thousands of base pairs. These discoveries have forced recognition that the genome is far less standardized than the concept of the human genome suggested. Each person's genome is unique, not just in SNPs, but in significant structural organizations. This explains some of the person-to-person variation in disease susceptibility and drug response that remained mysterious when analyzing only SNP variation. Epigenetics, the layer beyond DNA sequence. Perhaps the most profound recognition has been that DNA sequence alone does not determine the functional genome. The information content of genomes is massively expanded by epigenetic modifications, chemical tags attached to DNA and histone proteins that regulate whether genes are expressed without changing the underlying DNA sequence. DNA methylation, addition of methyl groups to cytosine bases, particularly at CpG denucleotides, is the most well-characterized epigenetic modification. Methylated DNA tends to be in a repressed chromatin state, with genes silenced. Unmethylated DNA tends toward active chromatin, with genes accessible for transcription. During development, establishing proper methylation patterns is essential. Disruption of DNA methylation through mutations in methyltransferases or through environmental exposures leads to developmental disorders and cancer. Histone modifications, chemical tags on the proteins around which DNA wraps, add another layer of complexity. Dozens of distinct histone modifications have been identified, each associated with specific chromatin states and gene activity patterns. A particular pattern of histone modifications marks active genes, while different patterns mark silenced genes or regulatory elements. During development, changing histone modification patterns across the genome drives cell fate decisions, how a multipotent progenitor cell becomes specialized as a neuron versus a heart cell versus a liver cell. The field of epigenomics has expanded explosively. Researchers can now map epigenetic modifications at base pair resolution across the entire genome. This has revealed that epigenetic patterns are not random but highly organized and precisely controlled. Epigenetic dysregulation contributes to virtually every complex disease studied. Cancer through altered methylation of tumor suppressors, psychiatric disorders through aberrant histone modifications affecting neurodevelopmental genes, and metabolic disease through epigenetic changes affecting metabolic regulators. Critically, epigenetic modifications can be dynamic and reversible, responding to environmental signals like diet, stress, toxin exposure, and exercise. This represents the mechanism by which environmental factors influence gene expression. A particular diet might alter DNA methylation patterns, affecting genes regulating metabolism and longevity. Chronic stress might change histone modifications, affecting immune genes, increasing disease susceptibility. This revelation has transformed biology from viewing nature and nurture as dichotomous to understanding them as interacting at the molecular level. Chromatin architecture and three-dimensional genome organization. Another revolutionary insight has been recognizing that the genome is not simply a linear molecule, but a highly organized three-dimensional structure. The discovery of this three-dimensional architecture emerged from techniques like Hi-C, which identifies physical interactions between distant DNA regions, and subsequently from advanced imaging of live chromatin in cells. Chromosomes within the nucleus are not randomly organized. Instead, they occupy distinct territories. More intriguingly, individual chromosomes display a complex, hierarchical architecture. DNA segments form chromatin loops, hundreds of thousands of bases folding back on themselves to create compact structures. These loops bring distant regulatory elements into contact with genes, enabling enhancers to control target genes. The proteins that hold these loops together, particularly cohesin and its regulators, are essential for proper gene regulation. Topologically associating domains, TADs, are megabase scale regions within which DNA sequences preferentially interact with each other but rarely interact across domain boundaries. TADs appear to organize the genome into functional units, with genes and their regulatory elements typically residing within the same TAD. Disruption of TAD boundaries, through structural variations or mutations in TAD-forming proteins, can cause disease by separating genes from their regulatory elements or inappropriately bringing regulatory elements into contact with new targets. This three-dimensional organization is dynamic. During development, as cells differentiate, TAD structures change. Different cell types display different chromatin architectures. Cancer cells often show profound reorganization of chromatin architecture, which contributes to their dysregulated gene expression. Understanding this architecture has proven crucial for interpreting genetic findings. A structural variant affecting disease susceptibility might not disrupt a gene, but rather disrupt the chromatin looping that brings an enhancer into contact with its target gene. Without understanding three-dimensional genome organization, such mutations would seem mysterious. The dark genome, sequences beyond standard annotation. Despite advances in genome annotation, significant portions of the genome remain functionally mysterious. The dark genome, sequences that show evolutionary constraint, suggesting they're functionally important, but whose specific functions remain unknown, comprises perhaps 5 to 10% of the human genome. Some dark genome sequences might encode proteins too short or too weakly expressed to be detected by standard proteomic approaches. Others might encode regulatory RNAs not yet classified. Still others might function through mechanisms entirely different from known regulatory paradigms. A particularly intriguing possibility is that some dark genome sequences function through non-canonical DNA structures. Recent research has revealed that DNA can form structures beyond the canonical double helix. Jigkey quadruplexes, four-stranded structures formed by guanine-rich sequences, occur in thousands of genomic locations, particularly near gene promoters and regulatory regions. These structures can be recognized by specific proteins and appear to have regulatory functions. zDNA, a left-handed DNA helix distinct from normal right. While handed B-form DNA occurs in alternating purine-pyrimidine sequences and also appears to have regulatory roles. The discovery of these non-canonical DNA structures suggests that genomic information is encoded not just in sequence, but in the physical structures that sequence can assume. This opens entirely new possibilities for how mutations might cause disease through disrupting normal DNA structure. The first inklings that this view is wrong emerged gradually. In the 1970s and 1980s, researchers discovered regulatory DNA sequences, stretches of DNA upstream of genes that controlled whether genes were expressed. These sequences were functionally crucial, but did not code for proteins. Throughout the 1990s and 2000s, evidence accumulated that many disease-associated mutations lay not in genes, but in these intergenic regions. Yet the significance of these findings was not fully appreciated until technologies emerged that could systematically probe the entire non-coding genome. The ENCODE revelation, the turning point came with the ENCODE project, Encyclopedia of DNA Elements, a massive international consortium that diverse experimental techniques to map which regions of the genome were actually being used by cells. The results were startling, using tools like ChIP-seq, which identifies where proteins bind to DNA, DNASI-seq, which reveals accessible open chromatin regions, and RNA-seq, which measures all RNA molecules produced. Researchers discovered that the majority of the human genome showed evidence of biochemical activity. When? Harbors far more regulatory complexity than anyone anticipated. Advanced techniques have revolutionized our understanding of this hidden architecture. Modern researchers distinguish between different categories of amplified gene activity from vast distances, silencers that repress gene activity, and non-coding RNAs that regulate virtually every cellular process. Perhaps the most transformative discovery has been understanding enhancers, DNA sequences that control how strongly genes are expressed. Enhancers are not located immediately upstream of genes in simple regulatory relationships. Instead, they can sit hundreds of thousands of base pairs away, yet still exert precise control over gene, each responding to different environmental signals, developmental stages, or cellular contexts. A gene might have one enhancer active in the heart, a different enhancer active in the brain, and yet another in immune cells. This hierarchical regulation explains how the same DNA sequence can produce radically different outcomes in different tissues. In 2028, researchers have cataloged hundreds of thousands of enhancers in the human genome. They've discovered that enhancer mutations, even single nucleotide changes, can profoundly alter gene expression and cause disease. Children with developmental disorders often carry mutations not in genes themselves, but in distant enhancers controlling crucial developmental genes. The same enhancer mutations that cause disease in some genetic backgrounds might have no effect in others due to epistasis, interactions between the enhancer and other genetic variants elsewhere in the genome. A striking example illustrates the importance of these regulatory elements. A specific enhancer controls the HOX genes responsible for limb development. Mutations in this enhancer can cause conditions like polydactyly, extra fingers or toes, or oligodactyly, missing digits, without altering the HOX genes themselves. These enhancer mutations demonstrate that understanding disease causation requires understanding the full regulatory architecture, not just genes. Enhancers, but have opposite effects. A single DNA region might contain multiple overlapping enhancers and silencers, creating complex regulatory logic where genes are controlled by the balance between activating and repressing signals. The universe of non-coding RNAs. Equally revolutionary has been recognizing that the genome encodes not just proteins, but an entire universe of functional RNA molecules. The central dogma of molecular biology held that DNA is transcribed into messenger RNA, mRNA, which is translated into proteins. RNAs themselves were viewed as transient intermediaries with no independent function beyond carrying genetic instructions. This view has been completely overturned. Today, in 2028, researchers recognize at least a dozen distinct categories of functional non-coding RNAs, each with specialized regulatory roles. MicroRNAs, miRNAs, are short RNA molecules, approximately 22 nucleotides long, that regulate gene expression post-transcriptionally by binding to mRNAs and preventing their translation or promoting their degradation. A single miRNA can regulate hundreds of different mRNAs, making these molecules fundamental regulators of cellular processes. Dysregulation of specific miRNAs drives many cancers. Conversely, manipulating miRNA levels represents a promising therapeutic approach. Long non-coding RNAs, LNCRNAs, transcripts exceeding 200 nucleotides that do not code for proteins, perform diverse regulatory functions. Some LNCRNAs serve as scaffolds for protein complexes. Others guide proteins to specific genomic locations. Still others compete for miRNA binding, thereby modulating miRNA function. The number of small nucleolar RNAs, SNO-RNAs, guide chemical modifications to other RNA molecules, particularly ribosomal RNA. These modifications appear subtle but prove functionally critical. Dysregulation of SNO-RNA-guided modifications has been linked to developmental disorders and cancer. Circular RNAs, circRNAs, molecules formed when RNA splicing joins the end of an RNA back to its beginning, were discovered relatively recently and are still being fully characterized. Evidence suggests many circRNAs function as miRNA sponges, sequestering miRNAs and preventing them from regulating their normal targets. This provides another layer of post-transcriptional regulation. The discovery that the genome encodes all these regulatory RNAs means that a vast portion of the transcribed genome serves regulatory rather than protein coding functions. Mutations affecting these non-coding RNAs can cause disease just as readily as protein coding mutations. A child with autism might carry a mutation disrupting an LNCRNA essential for neural development. A cancer patient's tumor might have amplified a circRNA that sequesters tumor suppressor targeting miRNAs, structural variations, and the missing heritability. Alongside understanding regulatory elements, researchers have recognized that the genome is far more structurally variable than the traditional sequence-based view appreciated. For decades, geneticists focused almost entirely on single nucleotide polymorphisms, SNPs, which are common genetic variants affecting individual DNA letters. Yet most humans carry hundreds of structural variations in their genomes, some inherited, some arising de novo. Many structural variations are benign, but others profoundly phenotypes. A duplication of a critical developmental gene might cause intellectual disability or autism. An inversion might disrupt a regulatory element and cause disease. Importantly, by 2228, researchers have realized that structural variations account for a substantial portion of the missing heritability that plagued genetic studies. And for decades,